Transfection is the next topic. Transfection, we were studying about recombinant DNA technology, that is bacterial transformation in recombinant DNA technology we have studied. Next is the transfection. What is transfection? Transfection is the transformation of non-bacterial cells. It is the transformation of non-bacterial cells. The transfection, in transfection we are introducing foreign DNA into the eukaryotic cells. So this foreign DNA should enter into a cell. For a cell, there is, in a eukaryotic cell, there is a plasma membrane, and inside there is the nuclear membrane. So to enter into the nucleus, this foreign DNA should cross two membranes. One is the plasma membrane of the cell and other is the nuclear membrane. And we can introduce foreign DNA into the eukaryotic cell. There are two strategies for transfection. One is transient transfection and other is stable transfection. The transient transfection, in transient transfection is only for a limited period of time. The introduced DNA or the foreign DNA will remain in the eukaryotic cell for only for 1 week, and after that it will be destroyed by the nucleases present in the cell. And depending upon the nature of the foreign DNA, and the transiently transfected gene can generally be detected for only 1 to 7 days. So transiently transfected cells are typically harvested 24 to 96 days, and these can be used for research purposes, or for expression of genes. So it is more expressed than stable transfection. That is called the transient transfection. Next is the stable transfection. In stable transfection, the transfected DNA, that is the foreign DNA, is either integrated into the genome, into the chromosome of the eukaryotic cell, or it can be maintained as an episome, a genetic element in bacteria that can replicate free in the cytoplasm, or can be inserted into the main bacterial chromosome and replicate with the chromosome. Here we consider the eukaryotic cells. It can integrate with the chromosome, or it can remain in differently and it can replicate with the chromosome. So long-term maintenance of exogenous DNA in the transfected cell is possible in stable transfection. So it can remain there and transmit to next generation. These genes are transmitted to next generations, multiple generations. So usually a single or few copies of exogenous DNA integrated into genome only. Only a few copies are integrated into the genome. So expression is low compared to transient transfection.